Welcome back to Anatomy and Physiology 1 Laboratory. I'm Kevin Tokoph, and in this video we're going to go over key concepts from exercise 4, which pretty much is histology. Histology is defined as the study of tissues, and for the most part we're going to be looking at microscope slides, or images of these tissues on microscope slides. All right, so let's go into some of the basics. Now, in general, there are 4 tissue types. They're listed here: epithelia or epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Now, we have separate labs where we look at muscles and neurons and the parts of the brain. We're going to be looking at these tissues separately in those lessons. So in this exercise, we're only going to be looking at epithelial tissue and connective tissue. Okay, so we have 5 epithelial tissues and 6 connective tissues. We'll look at epithelia first, and let's actually look at how we classify epithelial tissue. We can classify epithelial tissue in 2 ways. One is based on the number of layers of the cells, and the other is by the shape of the cells. So let's look at the shape. So if the cells individually are flat and irregularly shaped like this, but the key is that they're flat, we call those squamous cells. All right. If they sort of have a square-like appearance, because they're really 3-dimensional, but when you look at them on a micrograph image, they're going to look like a square. So if they look like this, they're not much longer than they are wide. They're cuboidal in shape. And if you have cells that are a lot longer in one dimension than the other, they look like almost like a pill, so to speak, or a column. They are columnar. All right. Now let's look at the layers, the number of them, and see how we can classify it there. If there's one layer of these cells, the term for that is simple. Okay, so for example, here's squamous cells, and there's just one layer of those. Therefore, this type of tissue would be called simple squamous epithelium. All right. The second way to classify the layers generally is stratified. So here we see that there are multiple layers of these cells stacked on top of one another. And so because each individual cell is flat, but there's multiple layers, we would call this stratified squamous epithelium. And you can follow the same pattern down here. Here I have cube-like or square-like cells, but there's one layer of them. So this would be simple cuboidal epithelium. Whereas in this case, the cuboidal cells are stacked in multiple layers, so this would be stratified cuboidal. Down here, we have one layer of columnar cells, so this would be simple columnar epithelium. These are stacked in multiple layers, so these would be stratified columnar. And in general, the ones we're going to be looking at in this class are going to be simple squamous, stratified squamous, simple cuboidal, and simple columnar, along with one other. And that's over here, which is pseudostratified columnar epithelium. This one is pseudostratified because, as we'll go over in just a few minutes, notice that these cells are not perfectly columnar. Some of them may look like pizza slices. So someone just had a pizza, they cut it up into slices, and kind of just threw the slices on top of one another, to where it might look like, in some cases, this cell where my mouse is might be a layer on top of this one, but it's not a true second layer, so it's pseudostratified. And they're generally approximately columnar shaped, so we call it pseudostratified columnar epithelium. So this is kind of the exception to the rule. But other than that, you classify them in these ways. In this course, we will not be looking at stratified cuboidal or stratified columnar epithelium. And additionally, instead of using a microscope directly in class, as you saw or will see, we're actually just going to be looking at images of these slides just for the purpose of time. Here's a typical diagram of epithelium and connective tissue. Here we have epithelial cells, and these, since they are cuboidal in shape and there's one layer, this is probably simple cuboidal epithelium. And always with epithelium, on one side we have just a physical space, usually an empty space. There could be a number of things in this space, but the physical space is called the lumen, typically. Okay? The side of the epithelial tissue that faces the lumen is called the apical surface. So this side of every one of these cells is called the apical surface. On the other side of the epithelial tissue, we have connective tissue. So all this in here, all these squiggly lines, these are connective tissues of some kind. The side of the epithelial tissue that faces the connective tissue is called the basal surface. And associated with the basal surface is the basement membrane, which is this solid pink line right here. So the basement membrane is sort of an anchor that anchors these epithelial cells or the tissue to the underlying connective tissue. So the basement membrane in some ways is like the glue that sticks those two together. And the side of the cells that faces the connective tissue is the basal surface. In here you have connective tissue itself. Here's some cells, and you'll typically hear me talk about this term when we get to connective tissue, the matrix. So the matrix is really just everything but the cells. So all this stuff in here, all this fluid, which is actually called ground substance, and then these fibers, which could be collagen or something like that, all of this, with the exception of the cells, all of this is the matrix. All right? Now let's go into the epithelial tissues, and we have 5 of them. And these names right here on this slide, which you have, these are the names that you would need to put on the exam to get full credit. These full names right here. All right, first we have simple squamous epithelium. So these are micrograph images of that. So for simple squamous, this is the easiest picture to look at. Notice that these cells are completely flat. These darkened spaces right here, these dark parts, are the nuclei. Nuclei tend to stain dark, but you see that these cells are flat. This is actually in the lungs. These are actually the nuclei of the squamous cells for the alveoli, which you'll talk about in anatomy and physiology too. Another common place that you can find simple squamous epithelium is in the lining of blood vessels. So if you look at this part right here where the lining is, I'm kind of tracing it with my mouse, you see that these cells are very flat. Here's one cell right here denoted by its nucleus. Here's a second cell, again denoted by the nucleus, possibly a third, a fourth, and you can see that these cells line the blood vessel. This is actually a longitudinal section of the blood vessel. Here's a cross-sectional view of it, and again you can see simple squamous cells lining the surface of that. And notice in here, in this space that doesn't stain at all, this is the lumen. Okay, here's the lumen inside here. So the lumen is, like we said, just usually a space, and things can be inside of it. For example, here blood runs through this lumen, and since this is also a blood vessel, blood runs through here as well. And so this part of these cells would be the apical surface, okay, and then the other side would be the basal surface. All right, so simple squamous, make sure you can identify these. All right, let's move on to simple cuboidal epithelium. A very common way, at least in this course, that you'll see simple cuboidal epithelium is you'll see these cuboidal cells, very large nuclei relative to the size of the cell, and they go around some lumen like this in a circular pattern. So notice these simple cuboidal epithelial cells form a circle around this lumen. So here's one system right here. Here's another clear system right here. Simple cuboidal cells running around this lumen. Here's another picture right here, and again right here. So you can see this setup is very common. These are cross-sectional views of this, by the way. Also notice just some more terminology. Again, here's our lumen. So this side of these cells is the apical surface, and not listed here, this would be where the basal surface is, but also associated with the basement membrane, which would be sort of here in these yellow dotted lines. All right. And also, in general locations, simple cuboidal epithelial tissue would be found in kidney tubules, the ducts of many glands, and the glands themselves. All right, so these are simple cuboidal epithelia. All right, let's move on to simple columnar epithelium. All right, so these are literally just column-shaped cells. You can see here, here's each individual nucleus, but notice here, we've got these elongated cells like this, okay? You can see again a picture of this here. Now, these cells are located mainly in the digestive tract and also excretory ducts of glands. And since they're located in places like the digestive tract where you need secretions of enzymes and mucus, you need specialized cells that are actually able to produce and release this mucus and so forth into tracts. So notice here we have other types of cells embedded in the simple columnar epithelium. And these are called goblet cells. And they typically appear as kind of clear spaces almost. Not completely clear, but they certainly don't stain as dark as the simple columnar cells. So these right here that I have labeled, these are very clear and eminent goblet cells. So in this type of tissue, if you're given this, you need to pay close attention to whether or not it says name the tissue or name this cell that I have an arrow pointed to. I may have an arrow pointed at the goblet cell itself, all right? And again, here's the lumen up here where my mouse is, okay? In this picture down here, this would be the lumen, all right? And there's not really super clear goblet cells, but I might guess this might here, since it didn't stain, this might possibly be a goblet cell, but these are definitely 3 right here, all right? Now, simple columnar epithelium is not the only tissue to have goblet cells. The other one is ciliated pseudostratified columnar epithelium. Kind of a pain to write that all out, but you do need to do that. Notice with these are pseudostratified, so these are not perfect columnar cells. Remember when we went to this picture, it looked as if it was very uneven. They're not perfect columns, and then also it might look like in some cases someone took the pieces of a pizza and just kind of threw them onto a microscope slide. You can tell these are definitely not perfect columns. Also notice here, these also have goblet cells. All right, so here's 2 right here, possibly a couple over here. We go down to this picture, here's a goblet cell. There's a goblet cell you can see labeled as G for goblet. There's actually quite a few in this picture down here at the bottom. Another thing to notice, and this is an easy way to distinguish between ciliated pseudostratified columnar epithelium and the previous one, is that these are ciliated. If you look at the apical surface of these cells, there are hair-like projections, very dense hair-like projections called cilia. You can actually see them in all 3 of these pictures. This one over here on the right, it's not as clear, but you can still see them on this darkened apical surface. It's easily visible in both this one right here and this one at the bottom. In fact, it's labeled as C for cilia. So if I pointed to these structures right here, you would need to name those as cilia. Notice that the previous one, simple columnar, does not have these cilia. You see this very defined apical surface, but no hair-like projections on it. And that's how it differs from ciliated pseudostratified columnar epithelium. This tissue is generally located in the respiratory tract, especially the trachea, and these cilia are actually able to beat up and push particles and bacteria out and prevent them from getting deeper into the respiratory tract and preventing them from getting deep into the lungs. All right, so these have cilia and goblet cells. Make sure you watch out for those. The last kind of epithelial tissue that we're going to look at in this course is stratified squamous. Now remember, the difference between simple squamous and stratified squamous is the number of layers. We still have flat cells, but in stratified squamous epithelium, these cells are stacked. This is a really good image here on the top. You can clearly see that these cells are flat in appearance, at least at the top, and there's definitely multiple layers, all right? And if you look here on this bottom picture, you'll also see we've got a ton of flat cells, again in multiple layers. Usually the distinction between simple and stratified squamous is pretty obvious because there's usually a lot of layers. All right, and pretty much the stratified squamous epithelium is going to be in any location where you're going to need protection from abrasion. So if you think about the lining of the esophagus, we constantly eat food, and so the food can scrape off these cells, but luckily since they're stratified, they can grow back in multiple layers. Also, obviously the mouth, the vagina can be abrased, obviously, and then the epidermis. Obviously your skin is in constant contact with lots of things, so very important to have stratified squamous tissue in that region. All right. Now we're going to go into the connective tissues. I think to some extent these are actually a little bit easier than the epithelial tissues. That's just my opinion. And in this course we have 6 of them. All right, we're first going to look at the 3 types of cartilage. The first is elastic cartilage. Now, a few things about cartilage that will help you identify it. So see these things right here where my mouse is? There's a ton of these. These are called lacunae, or lacunae is the plural, and the lacunae are spaces in which you have cells called chondrocytes. So I'm actually guessing these little pink regions inside the lacunae, those are the chondrocytes, but they're in a space called a lacunae. All right, you do need to be able to recognize those. In many cases, particularly in elastic and hyaline cartilage, I think they kind of look like a bunch of octopus suckers. So if you actually look at a real octopus's arms, it actually has a ton of suckers on it, and they kind of look like that. So if you actually recognize those lacunae, that tells you it's cartilage. Now the question is, how do you determine what type of cartilage it is? Well, in terms of elastic cartilage, there's two kind of general ways to look at it. The first way, which is kind of a not as good way, because there can be some exceptions, but in general it's usually true. In elastic cartilage, these octopus suckers, or these lacunae, are actually much more dense and they're more tightly packed. If you look how tightly packed they are in this image versus the next one, hyaline cartilage, you'll definitely see that in elastic cartilage, these octopus suckers, or lacunae, are a lot more tightly packed, more dense. All right? But the better way to tell the difference between elastic and hyaline is that in elastic cartilage, this matrix outside of the lacunae, so every space here that's not occupied by lacunae, is very rough looking in appearance. Even if you look down here, you can tell it looks like there's a lot of scratch marks, all this stuff. So in elastic cartilage, the matrix is a lot rougher. All right. Now, in terms of the location, we have elastic cartilage in both the ear and the epiglottis. We won't be looking at the epiglottis until Anatomy and Physiology 2, but you can obviously feel your ears. You hopefully know your ears are made of cartilage, but the specific type is elastic. And if you get confused, think E for elastic, E for ear, and epiglottis. All right. The key with elastic cartilage is we have the lacunae with the chondrocytes, and the matrix is rough. In hyaline cartilage, we still have these octopus suckers. We have these lacunae, which you do need to be able to recognize, but notice that the matrix is a lot smoother. If you look outside of these, this looks like a much smoother matrix than it does in elastic cartilage. And that's sort of the giveaway that it's hyaline cartilage, all right? Also in hyaline cartilage, generally speaking, these lacunae are not as tightly packed together. They can be a little bit more sparse, but you should still easily be able to see them, all right? Now, hyaline cartilage you have in your joint cavities. Most joints, with the main exception being the spine, your vertebral column, most joints, all the bones that articulate in that joint are covered in a type of cartilage, which you will see later is called articular cartilage, but its specific type is hyaline cartilage. So for example, if you look in your knees, many of you may know about the meniscus in your knee. It's a commonly injured tissue. That is actually hyaline cartilage. It's part of an articular cartilage deposit in your knees, but all your joints have it. You also have hyaline cartilage in the ribs, the costal cartilage, which we'll look at in a couple lectures. The nose, your nose is cartilage, that's hyaline. And then also, whenever you develop in utero, you actually don't start out with bone. You start out with an embryonic skeleton made of cartilage, specifically hyaline cartilage. And over time, it ossifies into bone. All right? The last type of cartilage also has lacunae. They're not as obvious. They look a little bit different, but you can still usually tell they're lacunae. Here's an example right here where my mouse is on the bottom image. You see here a chondrocyte surrounded in kind of a whitish space. All of these things are lacunae. You can see them up here at the top. Now, fibrocartilage, the lacunae look a little bit different than they did in hyaline and elastic cartilage, but in fibrocartilage, we kind of have sort of what are called striations. So striations are sort of when you have a general appearance of like fibers kind of all going in the same direction. Okay, so notice if you kind of look at it like wisps of a cloud, in this top image, the fibers or striations appear to go from up here kind of down here, if you follow my mouse. From up here to kind of down there. From up here to kind of down there, okay? In the bottom picture, they seem to be going roughly left to right or like right to left, whatever, horizontally. But in fibrocartilage, you do have these striations, which is very important to recognize. You have both the lacunae with the chondrocytes, but also these striations that generally go in one direction, all right? Now, fibrocartilage, we're going to see this in 2 lectures from now, but when we look at the pelvis, the 2 halves of it, which are called os coxae, are actually joined by a type of fibrocartilage known as the pubic symphysis. But the more important area where you have fibrocartilage is actually the intervertebral discs, which we'll look at actually in the next lecture in bones. Each vertebra is actually cushioned from the adjacent one by a fibrocartilage deposit called the intervertebral disc. All right? So that's the 3 types of cartilage. Now we've got 3 other tissues, which I don't think are actually as bad. These are actually a little bit easier. This is a very easy one to recognize, adipose tissue. This is body fat, all right? Now, if you look at these cells, each one of these white blobs is a cell, all right? And these cells, since they're fat cells, they hold a tremendous amount of fat. So they hold triglycerides, cholesterol, all sorts of lipids, just lipids in general. And this entirety of it, this white, is all lipid. Now, this is a good cell right here where my mouse is. See this purple dot kind of on the exterior by its membrane? See, it's surrounded by the membrane, which is in purple, but this really dark spot, that's its nucleus. With adipose tissue, there's so much fat inside the cell that's stored, so much lipid, that literally all the organelles, including this nucleus, are all pushed to the periphery of the cell. So you can't see the other organelles because most of them are probably too small, but they would all be pushed to the membrane. The nucleus you can see here, and again, it's pushed to the periphery to the cell membrane because the lipid droplet inside is so massive. All right. Now, individually, each cell here is called an adipocyte, but this whole thing is adipose tissue. All right, so again, these are really easy to recognize. I like to put this on the test because it's one of my favorite tissues to study, so make sure to watch out for these. And pretty much this is just going to be anywhere where you have fat tissue. All right. This type of tissue is called loose connective tissue, although I actually learned it as areolar, which is another name. It's areolar tissue. You can use either one of these terms, areolar tissue or loose connective tissue. I think in class most people actually use loose connective tissue. Maybe it was easier to remember. But in general, areolar tissue or loose connective tissue looks like a spider web. I included a little image down here from Return of the King, Lord of the Rings. This character gets tied up in a giant spider's web, and that's kind of what areolar tissue looks like. Notice you have all this web-like fibers, and they go in all sorts of directions. Here's another image down here. Now, areolar tissue is one of these tissues, kind of like where we looked at goblet cells and cilia or these lacunae, right? So, areolar tissue, you have some things you need to know. First of all, these very thick pink fibers, like right where my mouse is, that pink fiber that's kind of going like this, they're much thicker in appearance actually in this one, but these thick pink fibers, those are called collagen fibers. So they are collagen. Your textbook calls them collagenous fibers. You can use whatever term you want. Either one is correct. I always call them collagen. That's how I learned it. Also, you see these much thinner purple fibers. Let's see if I can find a good one. Here's one right here. They're kind of going up and down like this. This thin purple fiber, again, there's some down here. Looks like here's a good one right there. My mouse is in the bottom right corner. Those are elastic fibers, although the usual scientific term is elastin, but you can use elastin or elastic fibers, all right? And again, the whole thing just looks like a spider web. You also have cells in here. Here's a mast cell. That's a type of immune cell. We also have other white blood cells. But in general, all these cells, collectively, your book calls interstitial cells. So there's actually a few things to know from this. Obviously that it's areolar tissue or loose connective tissue, but also being able to recognize collagen fibers, elastic fibers, and then the interstitial cells, which are pretty much just all these cells in the tissue. All right, and you're typically going to find loose connective tissue under many kinds of epithelial tissue underneath it, very similar to the picture we saw here. Okay, let's go down and look at the last one, dense regular connective tissue. This is the last one we're going to look at in this particular course. Dense regular connective tissue looks very similar to fibrocartilage. Okay? Now in fibrocartilage we do have striations, and we also have them in dense regular connective tissue. All of these striations go in the same general direction. You can see here in this one on the bottom left, they're kind of going like this. In this one on the right, they're going left to right or horizontally. In this picture on the top, they're going kind of like this, but they're striations. You may see some cells here, but these are not chondrocytes in lacuna. All right, these are just simple fibroblasts. All right, notice that they don't usually have this white surrounding. You'll typically just see them as purple blobs. They won't be surrounded by this kind of white or lightish region around them. So these are not lacunae, these are just simple fibroblasts, all right? And because there's no lacunae, that's how you recognize these as dense regular connective tissue. All right. Now, dense regular connective tissue, one of the common places that you find this is in tendons and ligaments, which we're going to talk about briefly when we do muscle tissue after the first practical. All right. You typically are going to find this tissue where things need to be very, very strong. All right. So again, that's all of the tissues that we're going to look at in this course. Until we get to muscle and nervous tissue later on. But for now, you need to know these 11 tissues. Again, I'm going to say this one more time. When you put the names of these tissues on the exam, these lists right here, these are what you need to write, okay? Even if it's a pain, these 4 words written out, dense regular connective tissue, or in this case, ciliated, pseudostratified, columnar epithelium, you need to write all of those out. Okay. Another important thing to make sure that you do when you're studying these is don't memorize these tissues by their color, because the color that you see in the slide depends on the scientist who stained it. There are different stains that you can use for the same tissue, and so the colors may look very different. An obvious example of this is usually seen with the cartilages. Here the cartilage matrix looks kind of pinkish, but here it's very dark. Another example where it's typically observed is actually fibrocartilage. Here they're both kind of pinkish, but this one's much darker. But I remember when we looked at this in class, we had one where the matrix was blue. So don't memorize these based on the colors. Memorize these based on the features that each one of these has. All right? So this has been a lecture over histology, the study of tissues. Hopefully this video helped. I will see you in class.